My job for the Nabiheal project was to do the hemocompatibility testing for the materials and the solutions that we were provided. So the received materials and solutions were combined as said in the protocols we got, and they were placed firstly in the cell medium cultures in a 12 or a 24 well plates, and were left to incubate for some time. Afterwards, the samples were collected from each well, and they were combined with the blood that we collected from the specimens here on the institute, and also left to incubate for a couple of hours. And after that, we basically took the serum by centrifugation and combined it with the drop-rinse reagents and all other stuff, so we could measure it on the UV spectrometer. And by measuring, we got the absorbance measurements that we used to calculate basically the concentrations of the hemoglobin in each sample. And the hemoglobin concentration implies the level of the lysis of the red blood cells. And
we are now in our clean room. We have like a few instruments here. It's the most expensive ones. I need to say this. Okay, first one, and I use this a lot because it's very useful for nanomaterials characterization. It's called Zetasizer, and it measures like hydrodynamic diameter based on dynamic light scattering method, and also zeta potential based on electrophoretic light scattering method. So we can get information like hydrodynamic diameter of nanoparticles and also zeta potential, and this is very important information about our systems. Also, this is AFM microscope, like atomic force microscope, but it's automatic. So you can see, if you know about the standard AFM microscope, it's a really huge instrument, and our is like a smaller version, and everything is inside this. So it's based on we measure nanomechanical properties of our like cells or tissues, and based on this we can divide some properties, like we have a healthy tissue and tumor tissue, and we can diverse this to tissue, and based on this we can get like better and rapid diagnostic tool. So today we have like we have one measurement, pancreas tissue. It's tumor tissue. So we need to get more information about this tissue, so we can in the future we can see if this can be separated from healthy tissue or not. So this is very important, and in the future it can probably replace PhD pathology like diagnostic tool. So it's rapid, and it's it will, I think it will be more advanced than previous ones. This is plate reader, but more advanced than we have in our non-classified room lab. So we can measure here fluorescence, absorbance, phosphorescence, etc. And here we have a cell discoverer. This is high content microscope, and it's fluorescence microscope, but we can measure cells in the time. So it gives us really good results, and it's really important for us to see how our cells respond during the time. We have one centrifuge here, and also balance, it's over here. That's it.
So we are now in the cell culture part of the lab. We have here the incubator, the laminar flow cabinet, and right behind you can see our microscope. These are some of the essential devices where you cannot work in the in vitro testing without those. Here we have the flow field fractionation system with the multi-angle light scatter detector. This is basically the analytical method. I am not an analytical chemist, so I don't know quite a lot about it, but what they have told me is that it is similar to liquid chromatography, but it is the difference is in it does not use column, but it uses this flow field fractionation system where the molecules, or you can also use nanoparticles, which is why this is interesting to us, are separated on this membrane here and then detected on the multi-light scatter detector. Here what you can see is the cell sorter. So with this device, what we can do is we can use basic principles of flow cytometry to separate cells into populations that we would like to obtain or discard, and then we can tell the device to put one of the populations that we want to keep into our flask, into our tube or anything that we are using, and the other ones it will discard by itself. So that's why it is quite good. This is also the analytical part of the lab. What we have here is the UV spectrometer, and then also here we have fluorescence spectrophotometer. Those are used by our chemists to obtain physicochemical characteristics of nanoparticles that we use in our lab. And another not so interesting part of the lab, but it is essential. Here we have the part where we can prepare samples. So we have the shaker, which can be used in quite various modes. We have the pH meter to test pH of our solution, because it's also important for cell cultures, but also for nanomaterials. Centrifuge, thermostat, and this is the water purification system that we have. And this was the short tour of our lab.